So I want to turn to another poll question and keep going here with this concept of well-characterized and what we've learned from the products that have come before us, right, and that are on the market today even. So the next poll question is, are the most widely used forms, or I should say serotypes of AAV, for example, AAV2, 5, 8, and 9, well-characterized today? So you have 3 answers, and I got a little snarky when I came up with answer C. But you can say, yes, they are highly characterized, B, they're absolutely not, or C, pray tell, what does well-characterized even mean? Which I think is something that we all are struggling with in general in this space, figuring out how we define something, right? So while the folks are addressing that, and if anyone wants to share their own thoughts in the Q&A spot, please feel free to share your own thoughts and elaborate more on what your thoughts on well-characterized means. But in the meantime, you know, we talked a little bit about already the impurities, particularly full, empty, partial capsids, have become a much bigger consideration now. I think there's been whole events around the characterization of AAV, and FDA has become a lot more interested in knowing exactly what is in a partially filled capsid, right, and how much of your sequence is in there. You know, I'd love to understand from your perspectives, both of you, where you're seeing some of the greatest analytical innovations happening today in terms of the equipment. I know, Andrew, you've already mentioned mass spec. You know, are there other technologies that we really could stand to use or better understand how to use in relation to AAV today, as we're learning to better characterize some of the more common impurities?
I mean, what immediately springs to mind is I am in love with mass photometry, because when I started in 2012, it was difficult to do development because you would need considerable sample and considerable time in order to do AUC, analytical ultracentrifugation. And then you have SEC moles, you have anion exchange, you can kind of optimize that, but they need somewhat optimization. They're not so good at the partials, obviously. So mass photometry has been a game changer. Sorry, mass photometry has been a game changer for me in terms of process development.
Okay.
So that's the technique that we've acquired since I started and that I've been most impressed with.
That's great. That's great. Go ahead, Justin. What are you thinking?
Yeah, I'd agree with that. And I think there's a lot of around the, you know, DNA characterization technologies between next generation sequencing, DNA electrophoresis, DNA CGD that help give you better idea as to what the quality of the DNA you're producing. You know, I think the mass spec helps you understand the capsid to some degree. These are also trying to understand the what's being packaged within there. There's been a lot of methods traditionally looking for residual DNA through, you know, PCR-based methods, but the technology is advanced to give you other options to really understand your DNA population.
And would you argue that this technology is understood by the regulators as well? Because I know that's another big piece of this, right, is making sure that regulators are comfortable as well with how you're using some of the newer, right, even sequencing technologies out there. Do you feel that there's openness to these newer technologies?
I think the more we discuss, the more we show the value of them, the more openness there will be around these. So I think the more these technologies get out there, the more accepted they are. So these are coming more to the space now. So that's the path I think it's on, but I think these really do help. They really help you fundamentally understand your product. And I would think they'd be pretty well received as you can articulate that.
The understanding of your product, characterization, right? Just go deep. Anything to add, Andrew?
No, I would just agree with that, and I think there will be, I think it would be an easy kind of sell to talk to regulatory agencies if you want to increasingly understand your product with the new techniques.
Yeah, absolutely. Miles, what's up with the poll question here? Do we have some? Doesn't look like we're having some issues with our technology. Nope, no, there we go. All right. So, are the most widely used forms of AAV well characterized today? We've got 23. Wow. All right. We're split on yes and no. We've got an even split. 23% say yes, 23% say no. And 53% of the industry shares my my snarkiness and says, "Pray tell, what does well characterized even mean?" Do you guys have any knee-jerk reactions to the responses here?
I think this just speaks to the degree of innovation in the field with where we're at today, right? You know, coming into this and being here almost 8 years, I'd say that we know a heck of a lot more about these products than we did 8 years ago, and the assays that have matured a lot. They are used for standard you know, analysis. Now we're getting into deeper characterization and understanding what we can do and what the value of those are. So I think, to me, that's what it gets at, is and how far can you push that to be well characterized? And that's the point we're in. To me, it's a maturity thing. With where we're at in industry.
That's a beautiful point. Andrew, would you agree?
I would agree with that. It also depends kind of what hat you're wearing at the particular time. So my analytical hat, if I was with my analytical team, I might say, well, no, they're not well characterized. We need to do this and this. But with process development, we might say, I'm comfortable that this is good and we can take it into patients.
I think you just brought up a really important point too. It's the topic of quality is a challenging one, right? Because I think we all, depending on the functional areas, we can have very different definitions or comfort levels with different levels of quality, right? And figuring out how to move forward with that and get everyone on the same page is perhaps an operational discussion, right? Within the, you know, how you address that quality question is going to depend ultimately on where you sit. So I think that that's a really important point to bring up.